Okay, so today's lab is going to be about the angiosperms, also known as the flowering plants. Now, in terms of the flowering plant life cycle, there are a lot of similarities with the gymnosperms. However, now the female gametophyte or the ovule is basically going to be contained in an ovary, which we see in this diagram over here. So this is the entire ovary, and then inside of the ovary will be the individual ovules. And depending on the species, there could be one ovule, there can be 8 ovules, 10 ovules. Again, it just depends on the species, but essentially the ovules, these individual female gametophytes, are going to be housed in the ovary, which is basically housed on the sporophyte of the flower, which is this guy right here. Now, the male gametophyte or the pollen grain is going to be contained in anthers. So this is essentially the male reproductive structure. So again, here is the anther, and within the anther are individual pollen grains or male gametophytes. Now, inside the anther, these individual pollen grains are going to undergo meiosis, and then those pollen grains will eventually become a microspore. That microspore is then going to undergo mitosis and produce more pollen grains, or essentially young male gametophytes. And these young male gametophytes can be dispersed in the wind. They can also be dispersed through animals, typically through insects that are pollinating these flowers and will carry that pollen on their back. As they're traveling from flower to flower, those pollen grains will typically stick to the female reproductive structure, specifically the stigma of that female reproductive structure. And when it lands on that stigma, it's going to get a cue to produce a pollen tube. And that pollen tube is basically going to inject itself into the style and release the sperm and fertilize one of the eggs inside the ovary. And then once that sperm fertilizes the egg, they'll produce a brand new zygote and then an embryo, and then eventually it'll produce a new seed as well. Now the ovule, same exact thing. The ovule is going to undergo meiosis, specifically the megaspore mother cell within the ovule, just like how it was with gymnosperms. It's going to undergo meiosis and produce megaspores, haploid megaspores. The surviving megaspore will then produce your female gametophyte, which will then eventually produce your egg, which is then going to be fertilized by the sperm. You get a zygote, then eventually an embryo, and then a brand new seed. So that is essentially the gist of the flowering plant life cycle. And again, a lot of these steps do overlap with the gymnosperms. Okay, so the main thing that I want you to get out of this slide is that the ovule is basically the female gametophyte or the megagametophyte. And within the female gametophyte or the ovule, you have a megaspore mother cell, just like how you have that diploid megaspore mother cell in the ovule of the gymnosperms. That megaspore mother cell undergoes meiosis and produces 4 haploid megaspores. Eventually, those 4 haploid megaspores are going to undergo a series of different sets of cell division. I don't need you to know that part. The only thing that I need you to know is that at the end of it, after all of those series of cell divisions, it's going to produce 7 cells total with 8 nuclei. And I'll tell you why there's an extra nuclei now. So like I said, once the megaspore undergoes a series of multiple cell divisions, it'll produce 7 cells and 8 nuclei. Now those 7 cells are the 3 antipodal cells, which is at the top right here. These 3 antipodal cells have an unknown function. No one exactly knows what they are used for. We just know that it is housed in the ovule. But again, they have an unknown function. And then you have your 1 central cell with your 2 polar nuclei. And that is why there are 7 cells and 8 nuclei. So it's basically an extra nuclei. And it's because that central cell has 2 nuclei. And then you have your egg right here, which again is going to be housed in the ovule. And then your 2 synergid cells. Now, these synergids are really important because they essentially help guide the pollen tube, they aid in fertilization, and they also help the zygote develop into the embryo when fertilization occurs. And it makes sense that these synergids are actually going to be typically close to the micropyle, because remember that that micropyle is the small little opening in the integument, which is the outermost layer of the ovule. And the micropyle is what the pollen grain is essentially trying to find. So when the pollen grain lands on this micropyle, this small little opening, that pollen grain is then going to get a cue to produce the pollen tube, specifically the tube cell of the pollen grain. So if you think back to the gymnosperms, the pollen grain was made up of two cells. It was made up of the tube cell and the generative cell. The generative cell was the one that produced the sperm, and the tube cell was the one that produced the pollen tube, hence the name tube cell. So it'll produce the pollen tube. The synergids basically help guide that pollen tube to the egg, and the pollen tube basically carries that sperm, those two sperm cells, to the egg so that it can fertilize it and then produce a new embryo. So again, 7 cells with 8 nuclei, and there's an extra nuclei because that central cell has 2 polar nuclei rather than just 1. So one characteristic that unites all of the angiosperms, all of the flowering plants, is double fertilization. I talked a little bit about double fertilization last week when we were looking at the Nitales within the gymnosperms. Specifically, they are part of the conifer phylum. But with the Nitales, so remember that the generative cell produces 2 sperm. In the gymnosperms, only one of the sperm would fertilize the egg and the other sperm dies, so resulting in only 1 embryo. In this case, with double fertilization, both of those sperm cells that the generative cell produces are used. So one of the sperm is obviously going to unite with the egg. It's going to undergo fertilization and produce a new embryo. The other sperm cell is going to basically unite with the 2 polar nuclei in the central cell and form a triploid endosperm, so a 3N endosperm. This endosperm is essentially going to be the nutrients for the growing embryo. So you can think of this as the food tissue or the food supply that's going to provide those nutrients for that embryo. If you think back to the gymnosperms, it was the female gametophyte, which was just haploid, that acted as the food tissue for the embryo. Now in angiosperms, the food tissue is triploid, so 3 copies of the genome. When fertilized, the ovule will eventually develop into a seed with a seed coat. So this ovule will eventually become a seed, and you'll no longer have this integument. You will now have a seed coat once that's fertilized. So remember, the integument is the outermost layer, and the seed coat is also the outermost layer of that fertilized seed. But remember, it's derived from the integument of the ovule. Okay, so now moving on to the pollen grain, also known as the male gametophyte. So I did show you guys this image last week during the gymnosperm lecture, basically just to show you guys that the pollen grain is made up of 2 cells, which is the tube cell, again, that will produce the pollen tube, and the generative cell, which will produce the 2 sperm. And the pollen tube, once it gets produced from the tube cell, is basically carrying the 2 sperm cells that are produced from the generative cell to the egg. Any questions so far? Good? Okay, anybody in the chat have any questions? Feel free to unmute yourselves if you don't have your video on once again, or you can always type it in the chat and I'll get a notification if you guys type something in. Okay, but now we're gonna go into flowers and the different flower parts. So all flower parts are essentially modified leaves, and we have 3 different non-reproductive structures that we're gonna be talking about. So we have the receptacle, which is this part right here that my cursor is on, and it's basically the thickened stem material from which the flower part grows out of. So you can almost think of it as like an extension of the flower stem, which is the pedicel right here. But it's basically the part of the stem that the flower sits on. And then you have your sepals. So you can see that here in this diagram up at the top, and then right here on the underside of the flower, these would be the sepals. The sepals are the one that, to me, look more like true leaves, at least for a lot of dicots. Most of the time, the sepals are going to be green, but for example, some monocots, sometimes the sepals are different colors. So it could be hard sometimes to tell the difference between sepals and petals, but typically the sepals are going to be the outermost whorl. And then the petals, which are these right here in this diagram, are usually going to be the one that's really colorful. They might have some patterns to it, and that's the part that's what is going to attract the pollinators. The petals altogether make up what we call a corolla. It's basically just referring to the petals. And then the calyx is made up of all the sepals. So receptacle, sepals, and then petals. And these are all the non-reproductive structures. And then we have the reproductive structures. So the male structures, which are these right here, this elongated structure is what we refer to as the stamen. And the stamen is made up of two parts. You have your anther, which will house the pollen sacs, and those pollen sacs are going to house individual pollen grains. And then you have your filament, which is this elongated structure right here. And then if you look at this diagram, again, here's the anther at the top, and then here's your filament. The anther is made up of individual pollen sacs. Usually there's going to be 4 pollen sacs. Oops. And within these individual pollen sacs is where all the pollen grains are going to be housed. And then you have your female reproductive structure, which is right here in the center, also known as the pistil, and it's made up of 3 parts. You have your stigma at the very top, and remember that the stigma is actually the part that's usually going to be sticky so that the pollen can easily stick to the stigma. And then below your stigma is this elongated, almost neck-like structure, which is the style. And this is where the pollen tube is going to grow down into. And then you have your ovary at the very bottom. The ovary, remember, is what contains the individual ovules or the female gametophytes. When the ovary is mature, this will then become the fruit, and the ovules will then become the seeds that are housed within that fruit. So looking at this diagram here, as you can see, both the male and the female reproductive structures are on this same flower, so both the stamens and the pistils. If both the male and the female reproductive structures, again, the stamens and the pistils are on the same flower, then that is called a bisexual flower. So again, it will have both the stamens and the pistil on the same flower. However, a male flower will obviously only have the stamens or the male parts in one single flower, and then the female will obviously only have the pistil or the female parts in that one flower. So when you refer to a flower, or when you say use the terms bisexual, male, or female, that is typically referring to the flower type, versus if you're talking monoecious or dioecious, you are referring to the species as a whole. So dioecious species will have male plants with only male flowers on them, and female plants will have female flowers with only female flowers on them. Now, monoecious species will have separate male and female flowers on the same plant. However, this doesn't necessarily mean that all of them are going to be just male or female. Some monoecious species and dioecious species can have bisexual flowers as well. But just think dioecious, 2 separate plants, monoecious, 1 plant, both male and female flowers. Okay, any questions before I move into pollinators? All good? Okay, so just so you guys know, the specialization in flower form, or when I talk about pollinators, it's not strict at all, especially when I talk about the colors that attract certain pollinators. It's more of a general rule of thumb, because I've seen bees pollinate pretty much any kind of flower they can get their hands on. So again, general rule of thumb, it's not strict though at all. But with specialization in flower form, pollinators essentially co-evolved with flowers, selecting for certain traits. So long-throated flowers are typically for birds, bats, and moths. So with birds, they usually have that long beak, and that basically allows them to get into the nectar in that ovary right there. And then at the same time, subsequently, get some pollen, and then as they move to another flower, it'll spread out that pollen. And then the flattened landing pads, which is typical of a lot of asters, is typically for bees, flies, and butterflies. So anything that can essentially stand upright onto that flower. Now, in terms of colors, white will typically attract moths and beetles. Reds will typically attract birds and butterflies. Blues and yellows are typically for bees, but like I said, bees will pretty much pollinate anything. Orange, pinks, and purples will also typically attract butterflies. Reds are also very commonly pollinated by hummingbirds as well. A lot of flowers also produce nectar guides, which are basically these patterns on the petals that basically allow the pollinator, or kind of guide the pollinator inside the flower and help them get to the nectar a lot easier. For example, these foxgloves right here, they have these nice petals, or these nice patterns on their petals. Some insects can also, basically this is what we see with the flower, and then this is what they see under UV wavelength. And it basically helps them guide them to the nectar. Again, these colors though are not strict at all. Kind of a general rule of thumb. And then just some pretty cool pictures of different pollinators and the flowers they usually pollinate. So here's a bird pollinating what I believe is Justicia californica, not 100% sure, but again, birds will typically pollinate the more long-throated flowers, and they usually like the red colors as well. And then obviously the bees will pollinate pretty much anything, but again, they usually like flowers that have that nice flattened landing pad. And then I thought this was a cool picture. It's basically this rat climbing up and down this inflorescence. It's pretty much to show you that anything can pollinate any pollinators, or pollinators can pollinate anything basically. Again, not strict at all. And then we have the special birds of paradise. So basically this bird lands on these, I believe these are the modified sepals. When they land on those modified sepals, it basically opens and exposes the stamens and allows for the release of all of the pollen. So if you were to see a birds of paradise, which we have all over campus, pull this part back, and when you open it, you'll see a bunch of like little white fluff, and that's all the pollen. Okay, so now we're gonna go over the inflorescences. There are a bunch of different type of inflorescences, but we're only gonna go over 2 types. So one of them is the raceme, and these guys have indeterminate growth, meaning that they're just gonna continue to produce more and more flowers. And typically with racemes, the older flowers are going to be at the bottom. So usually notice that these flowers are a lot more mature. These are the ones that have opened already. And then the flowers at the top are typically the younger ones. So these have not matured yet. They're typically just going to be flower buds. And then we have our head or capitulum. This is typical for members of the Asteraceae or the sunflower family. And with the head or capitulum, there are 2 kinds of flowers. We have the ray flower and the disc flower, which are here in the center. Now, when you look at a sunflower, people often think that that entire flower is one single flower, but actually a sunflower is an inflorescence. So it's a clustered arrangement of multiple flowers on one stem. So actually this part that looks like a petal, this ray flower here, this is actually one flower. This is one disc flower, another disc flower, another disc flower, and so on and so forth. So sunflowers are an inflorescence. So they're made up of tons and tons of flowers that kind of conglomerate together. And the head or capitulum, again, is made up of 2 types of flowers. You have your ray flower, and then you have your disc flowers right here. In your disc flower, you can see that here's the stigma, and it will usually have fused anthers, and then here's your ovary all the way at the bottom. And then if you look at the ray flower, here's the stigma once again with the fused anthers, and then your ovary in the bottom. So usually the style will be right about here, but it's typically going to be hidden by the anthers. But yes, sunflowers are a conglomeration of multiple flowers. And fun fact, usually for the disc flowers, the ones to the outside, so like here labeled in red, these are typically the older disc flowers, which means that their seeds are going to be a lot larger. So when you purchase sunflower seeds, they're most likely from the older disc flowers because those seeds are typically going to be bigger since they're older. Okay. So now going into seeds, not much again that I'm really going to be covering with seeds. The main thing that I need you to know that when a seed germinates, the first part that's going to be produced is your radicle. So that's your first formed primary root. And then from the radicle, you'll eventually get the production of your cotyledons, which are these guys right here. Those are the seed leaves. And then once you get the production of your cotyledons, eventually you'll get a hypocotyl. So that's just all the portion directly below the cotyledon. And then your epicotyl is everything above the cotyledon or your seed leaves. And then here are your roots right here. Okay, any questions before I go into fruits? Am I going too fast? Just a little? Does anyone want me to go over anything again? No? Okay. No response, so I'm gonna keep going, but feel free to stop me whenever. Okay, so fruits. So With the fruit, remember, like I said, it's basically the mature ovary. So when the ovary matures, that'll eventually become the fruit of that flower, and inside the ovary are the individual ovules. And when that matures, those ovules will become the seeds of the fruit. Now, most seeds have a surrounding pericarp, which is basically the fleshy ovary wall. So typically the part, or most of the time, the part of the fruit that you eat is that fleshy pericarp. So the ovary wall that's surrounding those seeds. One of the more common fruits are berries, and actually the berries that you know of, like strawberries, blackberries, raspberries, those are actually not true berries. I'll talk about what type of fruits they are in a second, but true berries are basically when all or most of that pericarp, so all or most of that ovary wall is fleshy. So a good example is a tomato. There is this long-standing debate about whether tomatoes are a fruit or a vegetable. They are most definitely a fruit. They are the mature ovary of that flower. They are a fruit. So again, all or most of the pericarp is fleshy. That is the definition for berries. Another good example for berries are any one of the peppers, so jalapenos, bell peppers, those are all true berries. A banana is a true berry. What else? Basically, most fruits are true berries. Pomegranate is an unusual berry because it has like this almost leathery exocarp, so a leathery outermost layer, and a pretty fleshy mesocarp. But this is indeed a true berry. There are also different subdivisions of berries. One of them is a pepo. So this is, for example, a watermelon or a squash, cucumbers, zucchini, anything closely related to the Cucurbitaceae or the cucumber family is essentially a pepo. So this is a berry with a hard or thick rind and a very fleshy mesocarp. So in this case, with pepos, which again is still a type of berry, the mesocarp, so the tissue to the inside is the part that is fleshy and the part that we typically eat. And the exocarp is the part that's that really thick and hard rind that we usually try to peel away or cut off. Another good example is the wild cucumber, Mara macrocarpa. This is also considered a pepo. Again, it just means a berry with a hard or thick rind, so a hard or thick outer layer. And then another type of berry is a Hesperidium. So this is a berry with a really leathery exocarp or leathery rind. So that's pretty much anything citrus. So lemons, oranges, limes, grapefruits, any kind of citrus fruit that is a Hesperidium. Again, it is still a type of berry though. It's just a berry with a leathery outer layer. Okay, and then we have our drupes. You guys probably know these as the stone fruits. So a drupe is basically when there is a stony endocarp. Now this is not the seed, but inside of the stony endocarp, which is what you see in this diagram right here, is where the seed would be. So stony endocarp, fleshy mesocarp, and then you have your exocarp or the skin, which we still usually eat as well. But again, these are typically stone fruits. Peaches, apricots, cherries, almonds are actually considered a drupe. They are not a true nut. True nuts are dry fruits, which I'll talk about in a second, but almonds are also an example of a drupe. Avocados are an example of a drupe as well. So again, it's a type of fruit with a very stony endocarp. Okay, so now we're gonna go into what strawberries actually truly are. And they are I believe they're technically called accessory fruits, but we typically refer to strawberries, raspberries, I believe blackberries as well too, as aggregate fruits. So they are not a true berry. A true berry only comes from one ovary from one single flower, and it results in a fruit that has a very fleshy pericarp. But for aggregate fruits, it does come from a single flower. However, that single flower has many ovaries, and those multiple ovaries kind of aggregate, hence the name aggregate fruit, or they conglomerate together to form one fleshy fruit. So that is what a strawberry is. It is an aggregate fruit. And then raspberries, I believe this is actually thimbleberry, but they are also aggregate fruits as well. The one that is the only berry that's actually a true berry is blueberries. And then we have multiple fruits. So the difference between a multiple fruit and an aggregate fruit is that aggregate fruits come from one single flower with multiple ovaries. But with multiple fruits, it's many individual flowers with many ovaries. One good example of a multiple fruit is a pineapple. There's actually a picture of a pineapple on the Lab 12 slides, but again, black mulberry, also considered a multiple fruit. Pineapple, a multiple fruit. If you look closely at a pineapple, actually, I have a pineapple. Let me grab one. Hold on. Oh, I forgot. I had a pineapple. Okay. So if you look at a pineapple, hold on. Okay. So if you look at a pineapple, you guys see these individual chambers? Okay, so this individual chamber basically came from the ovary of one flower. This is the ovary of another flower, so on and so forth. So technically this one chamber is one fruit, this is another fruit, and this is another fruit. Again, they come from multiple individual flowers that come together and form this one structure, the pineapple. With aggregate fruits like strawberries, those come from one single flower, but that one single flower has tons of ovaries. Does that make sense? Okay, cool. Okay, so now we're gonna go over dry fruits. There are two types of dry fruits. We have the dry dehiscent fruits and the dry indehiscent fruits. The only thing that I need you to know about these two types of dry fruits is that dehiscent fruits will split open at maturity and fling their seeds outwards. So these are some good examples of dry dehiscent fruits. Legumes or peas, cabbage, iris fruits are dry dehiscent fruits. Foxgloves, so those are the flowers that I showed you guys earlier. Poppies, those are all dry dehiscent fruits. So eventually they're gonna split open. Sometimes they'll have one split down the middle, sometimes they'll have two, but basically they're gonna open at maturity to allow for their seeds to be dispersed. Now, indehiscent fruits do not open at maturity. You typically have to force them open to release the seeds, like hazelnuts, for example. These are usually going to be carried by animals, sometimes wind, maybe water, but indehiscent fruits do not open at maturity. Dehiscent fruits will open at maturity to disperse their seeds. Okay, so now we're going to go over the three different groups within the angiosperms. We have the basal angiosperms, and then we have the monocots and the eudicots. Now, the monocots are actually technically a basal angiosperm, but they have such different characteristics that they kind of were made into their own group, but they are technically basal. And the eudicots are the more derived group, so they evolved much later. And some of the unifying traits of angiosperms is that they have a triploid endosperm, so a 3N endosperm. Remember, that was a result of double fertilization, in which all angiosperms have. Except for the Natales, they do have double fertilization as well, which is why they were thought to be the sister lineage to the angiosperms. The only difference with the Natales is that only one of the sperm is used, where in angiosperms, both of them are used. One of them will produce the embryo, and the other one will produce the triploid endosperm. And then with angiosperms, obviously they have the evolution of fruit, flower, and then the ovary / carpel, which is basically your female reproductive structure. Okay, so everything here in green are all of the basal angiosperms. I'm not going to talk about them in too much detail. If you want to learn more about the basal angiosperms and these individual groups, you should take Evolution of Plants, which I believe is going to be offered in the fall. But after the evolution of these basal angiosperms, we have the monocots, also known as the monocotyledons, which, like I said, they are technically a basal angiosperm. And then we have the eudicots, which is the more derived group. And again, the unifying characteristic for all angiosperms is the triploid endosperm, again, a result of double fertilization, and then the fruit, flower, and then the ovary, which is basically going to be your fruit. Okay, so the basal angiosperms are the first flowers that were animal pollinated. They have many flower parts, and because you usually can't tell the difference between the petals and the sepals, they have undifferentiated whorls of what we call tepals instead. So because again you can't tell the difference between the sepals and the petals, we'll typically refer to them as tepals instead. A lot of monocots have tepals as well because it is hard to tell the difference between the petals and the sepals at times. So again, we usually just refer to them as tepals. They do have helically arranged parts, and they usually will also have an indeterminate number of stamens. So that means they're just going to produce tons and tons of stamens, and then they'll have their one pistil typically in the center. So some examples of basal angiosperms are nymphaea, or the water lilies, magnolias, liriodendron, which can get pretty big. Other good examples of basal angiosperms are actually avocado, Persea americana, and then the California laurel bay. So Umbellarea californica is also a basal angiosperm. Okay, so now we're going to go into the monocots, also known as the monocotyledons. So like I said, these are technically considered basal, but with basal angiosperms, actually I should have mentioned this earlier, like I said, they're helically arranged, but most of the time their petals or their sepals, they're usually going to be arranged in parts of 6, so in flower parts of 6. Whereas monocots, they're typically going to be organized in 3s. So usually 3 petals, 3 sepals, usually nothing more, nothing less. One characteristic that is common for all, if not most, monocots is that they'll usually have one pollen furrow, so one basically pore, one dip in their pollen grain. Dr. Bobich has a really good picture of that in the Lab 12 slides if you need help visualizing that, but basically it just means that they have one pore in their pollen grain. They also have parallel leaf venation for the most part, so basically their veins do not branch out often. Oops, typo here, but they have one cotyledon or one seed leaf that is produced when the seed germinates, and then their vascular bundles are typically going to be arranged in an atactostelic arrangement. So that means that their vascular bundles are kind of randomly arranged. There is no true central pith, unlike with eusteles. Okay, and then for the eudicots, one characteristic that kind of unites the dicotyledons is that their pollen has 3 pores rather than 1. So with the monocots, the characteristic that kind of united them is that they all had one pollen or one pore in their pollen grain. Again, with eudicots, they have 3. So the eudicots evolved much later. This is the more derived group in the plant phylogeny. Their flower parts are organized in either 4s or 5s, whereas monocots were in 3s. They have tripartite pollen, so 3 furrows in their pollen grain, and they have net leaf venation. So usually with net leaf venation, they'll have one main midrib, and then that one midrib will kind of branch off into other lateral veins, and those lateral veins will continue to branch off, so on and so forth. I'll show you guys what that looks like in a second. But eudicots produce 2 cotyledons or 2 seed leaves, hence the name dicotyledon. And then eudicots will typically have eusteles as well. So that's when their vascular bundles are arranged in a ring, and they have a true central pith, and they also have pith rays as well. So this table kind of sums up all the characteristics that I just talked about. Monocots have 1 cotyledon, hence the name monocotyledon. Dicots have 2 cotyledons. For monocots, their floral parts are usually in 3s, so 3 sepals, which is typically going to be the outermost whorl, and then 3 petals. For dicots, their flower parts are in 4s or 5s. Monocots will typically have parallel leaf venation. So these little black lines that you see, if you follow my cursor, these are going to be the veins, and they're usually parallel to each other, hence the name parallel leaf venation. For dicots, they usually have net-like leaf veins. So you'll have one main vein or one midrib going down the center of the leaf, and then from there, those will branch off into lateral veins, and then these lateral veins will continue to branch off. So again, net-like leaf venation. Monocots have pollen grain with 1 pore or furrow, so there would be the 1 pore in the pollen grain, and then dicots will have 3, also known as tripartite pollen. And then keep in mind, monocots also have an atactostelic arrangement. So that's when their vascular bundles are arranged kind of randomly all throughout the stem, and they do not have a true central pith, but this is still all parenchyma in between those vascular bundles. Now with dicots, they will have a eustele, which means that their vascular bundles are arranged in a ring, and they also have that true central pith. They have the pith rays, which again is basically just the parenchyma in between the individual vascular bundles, and then cortex to the outside of those vascular bundles. Okay, so this is just showing you once again the difference between the atactostele, which again is typical of monocots, and the eustele, which is typical of dicots. So again, with the monocots, vascular bundles are arranged randomly, that is an atactostelic arrangement. No true pith, but this is still all parenchyma in the stem. And then with the eudicots, this is something that you're probably more familiar with when you think back to primary growth, which was weeks ago. But here's your central pith, which is mostly made up of parenchyma. And then you have your individual vascular bundles arranged in a ring. In between your vascular bundles are your pith rays, which again is just parenchyma. So your vascular bundle is always xylem to the inside, phloem to the outside, and then each vascular bundle will always have a fiber cap. And then to the outside of those, you have your cortex and then your epidermis, which is still your outermost layer. Okay, and then for the monocots, obviously they only produce one cotyledon, which is right here, but here's your embryo, your one cotyledon, and then your triploid endosperm. And then for your eudicots, like a radish seed, they'll produce two cotyledons. Again, that's why it's called a dicot, and why monocots are called monocot, because they only produce one seed leaf. And remember that monocots will typically have an adventitious root system. So that means that their radicle, their first formed primary root, is short-lived. So, and then for the taproot system, which is typical of dicots, they have their primary root or their radicle is long-lived. And all the roots are going to emerge from that one main taproot. Okay, so that was it for my lecture. Does anybody need me to go over anything again? Any questions? Felt like that was a little bit fast, so feel free to ask me if you want me to go over anything again. This is recorded too. No? Okay, well, if there are no questions, I'm gonna go ahead and stop this recording really quick.